Oh, that's it. We are on.
You're online?
Yep.
Okay, hello everyone. First of all, I will say a few words in Portuguese just to our students here. Pessoal, seguinte, a palestra de hoje é a primeira das nossas palestras internacionais com o Dr. Luiz Haro. Obviamente que a gente gostaria que vocês participassem. Então, assim, se vocês não se sentirem confortáveis em fazer perguntas ao Dr. Luiz em inglês, vocês podem fazer em português normalmente, podem ligar o microfone, imagino que o Luiz até entenda, se vocês fizerem a pergunta em português, mas, assim, a gente pode também traduzir, se vocês quiserem deixar a pergunta no chat, nós traduzimos para ele, caso ele não entenda alguma coisa, nós estamos aqui para ajudar, mas não deixem de participar simplesmente por questão de língua, porque aqui a gente está num ambiente super tranquilo, o Luiz é amigo nosso, então, assim, tá tudo em casa aqui, então vocês podem ficar tranquilos, tá certo? I will switch to English now to make the formal introduction of Dr. Luis. So, hello everyone, everybody that is there in our following this seminar on the YouTube. I was just saying to our students that we can interact with Dr. Luis either using English or Portuguese. If you want to let your question to Dr. Luis by the end of the seminar in Portuguese or in English in the chat, feel free to do it. Just please don't miss interacting with him because of language barriers. So we are here to help. We have actually several foreigners, so we can understand a lot of languages. We have Argentinians, and so we understand a lot of languages here. So language is not a problem. Then we can move to our seminar today. Today is our first international seminar. I would like to thank Dr. Luis de Haro for joining us here today for a very nice seminar, I'm pretty sure. Dr. Luis is a postdoc at the Weizmann Institute of Science in Israel, right? Luis, thank you very much for accepting our invitation to give a talk in our course today. In the name of the whole team, Magda, Leandro, and I, we are very pleased and thankful for your acceptance. So I will, normally here we have the tradition that Leandro is the person who introduced the speakers. So I will give the word to Leandro, and I already thank you very much for accepting this invitation.
Thank you, thank you.
Right, so Luis Alejandro de Haro was graduated in biology and master and PhD at the University of Buenos Aires, Argentina. Luis had a postdoc research fellow at the National Institute of Agricultural Technology, and now, as Igor said, is a postdoc at the Weizmann Institute of Science in Israel, working with the identification of novel genes associated with cannabinoid composition. Luis, thanks so much again for accepting, and so the students is yours now.
Okay. No, thank you again for inviting me. So I will start the presentation. Window here. Do you see my presentation?
Yes.
Yes.
Good. Okay. So, okay, thanks again, Magda and Leandro and Igor for inviting me to this talk. When Magda asked me to give this lecture, I asked her what should I talk about, because I've been here in Israel for about a year and a half, and all my work is unpublished yet. So I didn't want specifically to talk about this because it's unfinished work. So I decided to speak about the general approach that we have in the lab, or how I think we try to tackle the things here. And that's how I call the talk, Strategies for Genome Mining Towards the Identification of Novel Genes Involving Specialized Metabolites. And yes, I work in Asaf Aharoni's lab in the Weizmann Institute of Science in Israel. So first, I would like to show you, these are two pictures of the institute. The institute is located in Rehovot, which is the middle part of the country. We are half an hour from Tel Aviv, the capital of Israel. And as you can see, it's a very beautiful place, both architectonically speaking and also naturally speaking. It also takes a lot of effort because here there is not a lot of water, but it's a very, very beautiful place. So I recommend you that if you ever come to Israel, to visit the institute, which is open to the community also. So you are more than welcome to come. And I work in this lab, Asaf Aharoni lab. This one is Asaf, my PI. And we work on plant metabolomics in general. So the core of the lab is plant metabolomics. For this, we have a huge, very big lab with a lot of machines that are mass spectrometers and different machines that allows us to measure all kinds of molecules. And also this is accompanied by a group of very well-trained chemists that can develop different methods to detect almost any molecule that you can find in nature. Well, it's a bit an exaggeration, but we can detect a lot of molecules. And plants and kings of producing small molecules. As we are human and we need to classify the things. Usually, the classification in plants it's separated in terms of molecules are separated in primary metabolites, secondary metabolites, or specialized metabolites, and hormones. Primary metabolites are usually associated with those molecules that are involved in energy processes and growth and development. Secondary metabolites are more involved in interactions with other organisms or in adaptations to different kind of situations. And hormones are signaling molecules or involved in more regulation of different processes. But all these three categories, in most of the times, they mix each other, and a molecule that first was assigned as a secondary metabolite then will be involved in an energetic process. So it's this division is more for humans than the real thing. So the lab is mostly working on specialized metabolites, these molecules that are involved in interaction with other species, or in special adaptations to the environment. And these specialized metabolites are very, very important for humans because they can be products itself. They can be molecules that can work in medicine as anti-cancer molecules, anti-parasites, or psychotropics like cannabinoids or other molecules. And they can be also products in nutrition, like sweeteners that are non-caloric molecules that make that can be used for sweet to make things sweet without giving calories, and also vitamins, and also in the food industry as color dyes, like in ice cream or in any food that you want to dye with a natural colorant. You can use different molecules. This in this picture you can see a kind of Yes. These are betalines, which are a family of molecules that which pathway was discovered in this lab. And sometimes these molecules are not products itself, but are affecting agronomical traits. So they can be involved in abiotic stresses or trophic interactions, or they can be anti-nutritional agents, which are these molecules that produce problems for digestion or the incorporation of nutrients. Usually, when a plant is domesticated, these anti-nutritional agents are not there anymore. But like in potato, when the potato sprouts, then this sprout is full of glycoalkaloids, which is an anti-nutritional agent. If you try it, it will be bitter. So the general question of the lab is how and why the specialized metabolites are produced in plants. So a regular pipeline in the lab, this is very general. Of course, each project will have different its particularities, but this is like the regular pipeline that maybe can be generally applied for all the projects. And it is first about the metabolite. You find an interesting metabolite either because it's a product or it is involved in a particular process that you want to study. So first, what you have to do is to find a way to measure it. And for this is that we have all of these machines that can measure many things. And you start to ask yourself, okay, can I measure only this metabolite, or I can measure all the whole family? Usually metabolites are not the metabolites are a family of compounds that are related chemically. So you have to ask yourself, which type of instrument do I need? Do I need to develop quantitative methods, or I can manage with relative quantification? So then you have to ask yourself which plant species can produce it. If it is a specific species, or it's a family, a group of plants that can produce it, if they belong to the same family or not. And of course, if you can work with the plant to transform the tissue or to express genes transiently for infiltration or for during using BIGs, infecting the plant with a virus. So then you have to start profiling or getting more in deep about when and where this compound you can find it. Is the compound accumulated in a specific organs? It's in the whole plant, and the accumulation changes in different developmental conditions or hormonal treatments. And then once you have all of this more or less tackled, you have to start asking yourself to identify these new genes, which are the pathways involved in the biosynthesis of the metabolite. First, if there are genomic resources available for this plant, if there is a genome. If there is no genome, can I sequence the genome, or if there is a related sequence, can I do a resequence of the genome? Then can I do de novo transcriptomics, which is easier than doing the genome? And then once you have everything, you can try to find ways to find new genes or genes that are involved in this pathway, either by similarity to known genes or by co-expression. I will talk about this more in depth later, or if they are in gene clusters, which I will also talk in depth later. And finally, which is I think the most important part of the work, and it is also the most complicated. How can I check if this gene is actually involved in the pathway? So to do this, you can clone the gene and try to express it in different systems, in different microorganisms like E. coli or yeast, or you can do agroinfiltration, or you can try to do transgenic plants, either from the same species you are working or a model species that is easy to transform. And something I didn't put in this slide, but it's also super important, which is try to understand which is the evolutionary significance of the pathway. Why this metabolite exists? Why it exists in this specific plant, or it exists in different plants, in different families. So and then you have a full package, a full work. So as I said you before, what you have to do first is to develop a method to quantify the metabolite that you want to study, and for this we use mostly mass spectrometry. Mass spectrometry, first you have to separate the compounds, because if you do an extraction of a plant, of any tissue, you will find a mixture of thousands of molecules. So first you have to separate them using chromatography, and there are different types of chromatography that you will use depending on the nature, the chemical nature of the compound. But basically, or mostly, we use two types of chromatography, which is liquid chromatography when you have in the mobile phase a liquid, or gas chromatography when you have a gas in the as a mobile phase. And usually, gas chromatography is used if the compound is very less polar, and liquid chromatography is used if the compound is more polar. So after separating the compounds using this chromatography, you inject these separated molecules in a ionizer. The ionizer is a very high source of energy that will convert the molecules in charged molecules. So these charged molecules will go in a magnetic field that can be different with different. There are many different magnetic fields that will allow some of the molecules to reach the detector and some other molecules not to reach the detector. So you filter, you continue to filter the complexity of the mixture, and then. This, if you see the trace here in violet, is the injection of the different molecules. Each of these peaks is composed of different molecules that are separated using the chromatography, and then each of these peaks, when they are fragmented by the ionization agent, you get a spectra, and this spectra allows you to identify the compound, because this spectra is characteristic of the compound that is being destroyed. So, and to match this spectra, you have to either have the compound purified, you have to have a standard, or you have to purify it and you do an NMR. So to get the standards, we work with a few companies that are very, very big, and these companies, what they do is to basically to purify any compound that they can find in the nature. So then you have the standard to compare if the molecule that you think it is this molecule is actually the molecule that you think it is. And if there is no standard available, and this can happen in many cases, you have to purify the molecule and make a structural confirmation by nuclear magnetic resonance. And this is done also here in the institute by some very well-trained chemists that work here in our group, but also in other groups. So to identify the new genes, you have to have diversity. Diversity either both in the genes and in the metabolites. So you can have diversity in tissues. Here you have a picture of Arabidopsis, and it's the expression of genes in this case. But of course, the metabolite that you're studying will have different concentration in each of the tissues. And also you can study the diversity within tissues. Here, what I am showing you in this picture, do you see my mouse?
Yeah.
Good. In this picture, what I'm showing you is mass spectrometry imaging, which is a technique that we use a lot in the lab, and is basically to have a fixed tissue, and this fixed tissue is excited by a laser, and this excitation releases molecules that are injected in a detector. So basically, you can spatially detect different molecules within the tissues. So this way, you have diversity in the accumulation of the metabolite. And why diversity is so important? Because usually genes and metabolites are co-accumulated. So, and what this is in this picture, what I show you is a gene which has this expression pattern. Here you have different samples that can be different tissues or different conditions, and you can measure how similar they behave. No, it doesn't matter how the absolute concentration of the genes or the metabolites, but you can see how they change over time or over conditions, and you can measure this similarity in change. So this number means how similar these two genes behave. Okay, so if you have known metabolites that are the metabolite that you are studying, or you have known genes that you already know for other pathways, similar pathways, or pathways that were described in other organisms, you can have what we call baits. You use these genes to fish new genes using this measure of similarity. Okay, so this tool, we use it a lot to identify new genes, and for this we have to use a lot of transcriptomics, of course. And to use transcriptomics, I don't know if should I explain what is transcriptomics or Rna-Seq?
No, okay. I think it's not necessary. Just mention is a new generation sequencing, but I think everybody is more or less
okay. So
aware.
Okay, good. So here in this picture, what I am showing is the expression of genes, different genes or metabolites in different conditions or different tissues. So, and the one in red is a new gene that was identified only by measuring how similarly it behaves with the other one. So to do transcriptomics, usually you need a genome, because you have to align reads to a known reference. But this is also changing because the cost of sequencing is decreasing like crazy. Every year it's going lower, and not only the cost but also the quality of the sequencing. This graph, what maybe you saw it before, it's a very well-known graph, shows the decrease in the price over the years of the megabase of sequencing, and how it compares to the Moore's law, which is a prediction of how the microprocessors will increase its capacity over the years. So it's dropping a lot, and as I told you, it's not only the cost but also the quality. What do I mean with the quality? The length of the reads. Illumina, which was the company that allowed this to drop the price so much, the problem with Illumina is that it produces very short reads. So if you want to do a genome, it's complicated to do a genome with short reads because sometimes you have repetitive regions that doesn't allow you to assemble a genome. But now also there are new technologies like nanopore and PacBio, which are much longer and allow you to work with to do genomes much cheaper than before. I will talk about this a bit later. So to do transcriptome assembly, it's also relatively easy. This is just a picture to show a regular pipeline for transcriptome assembly and annotation. Basically, you extract RNA from different tissues of a plant or different conditions even, and then after you extract RNA, you sequence the RNA using Illumina. You do de novo assembly of these sequences. For this, you can use Trinity, which is a very well-known software, very validated, and then you can do annotation of all these transcripts, and this is relatively cheap and easy to do. And we did it a lot in the lab for different non-model species. So it's not so important to have a genome if you want to discover new genes. So using these kind of approaches, the lab has tackled many different metabolic pathways that were unknown or that were at least partially unknown. Mostly the lab works in sterol-like glycoalkaloids, which are one of these anti-nutritional agents that I was talking you before. Then, as I told you before, the lab also described the biosynthesis of the phthalines that are color molecules, very important to use in the food industry. Also, pipicolic acid, which is a molecule also involved in regulating plant defense and growth, and triterpenoid saponins. And I will talk specifically about this paper because I think it's a very good example of how we do things in the lab. This is a paper of Adam. He's a chemist by training, so he started to work. He wanted to understand how was the biosynthesis of these saponins, triterpenoid saponins, and the skeleton of the saponins is like you see here, and in the residues you can have a lot of decorations, a lot of different molecules that will make many different molecules. So and this family of molecules are very important for humans because you can get this one is an anti-inflammatory molecule, isin. QS21, it's an adjuvant for vaccines, and it's also particularly important now. And glycyrrhizin, it's a sweetener, which is extracted from licorice. And all of these molecules, there is no way to producing up to now, if it is not extracted from the plants. And the biosynthesis of them is not completely known. So Adam selected to work with spinach. Spinach is a member of the Caryophyllales family, and this family includes many edible plants that produce these triterpenoid saponins, and the biosynthetic pathway was unknown at this point, but the sequence was at that time was just being published. So what Adam did first was to extract and profile all the triterpenoid saponins. He did NMR for all of these, of the structure of all of these molecules. NMR means to purify the molecule and to know exactly the structure. And he named all of these saponins as iosocyte. So to start getting into the biosynthesis of these molecules, first he identified two genes that were previously known to produce this core structure, the aglycon structure, SOAP1 and SOAP2. These were identified by homology, just doing blast. But then, when he looked into the structure in the genome where these genes are located, he found the third gene of the pathway because it was very close to SOAP2. Many genes involved in secondary metabolites are located very close in the genome, which is known as metabolic gene clusters. So after this, he could say that he identified 3 genes. Of course, he tested biochemically this using yeast and agroinfiltration in Bentamiana plants. And then he did RNA-seq of different tissues of spinach. And using these 3 genes as baits, as I told you before, he could fish genes that were co-expressed with these 3 genes. So this is like telling you the end of the story, but after he could manage to identify all the saponins, he knew which was the nature of each of the enzymes that he was looking for. So he knew that SOAP2 to 4 were hydroxylases or oxidases, and so he tested different hydroxylases and oxidases from the co-expression data, and he found which ones were the ones responsible for this part of the pathway. And then he, using BIGS, viral induced gene silencing, it's a technique where you use a recombinant virus to infect the plant, and this recombinant virus has a fragment of the genes that you want to silence, and so you reduce expression and you see the phenotype of this reduced expression. So this way he identified SOAP6, SOAP7, SOAP8, and SOAP9, and this was relatively easy because he knew that it was glycosyl transferases. If you see here in this picture, you see this is a sugar, this is another sugar, this is another sugar, and this is another sugar. So he knew that he was looking for glycosyl transferases. But there was, and also he identified the last one, the last enzyme, this one, SOAP10, because he knew that it was an acyl transferase, and he tried 5 different acyl transferases that were that were identified in the co-expression data, and he identified this SOAP10, which was the last one. But there was one missing, that none of the co-expressed enzymes was compatible with the function that it was doing, and it resulted to be a cellulose synthase-like, which was not expected at all. So then the core of the paper changed, because he discovered a new function of an enzyme that was not expected before, and it was a part of the pathway that was missing in all the previous in all the previously described saponin biosynthesis. So he searched for this for homologs of this SOAP5 cellular synthase-like in different organisms, Fabaceae and Coreophyllales, and he identified and cloned these homologs of SOAP5, and he managed to to produce this compound using all of these enzymes. And finally, this sweetener that I was talking to you before, glycyrrhizin, which was extracted from a plant, also he managed to produce it in yeast using this enzyme. So, and what happens if your species doesn't have a genome? As I was telling you before, not only the prices of the sequencing are dropping, but also the quality of the sequences are increasing. And for this, there are two technologies. One is nanopore, and the other one is PacBio, or two companies that are working on this. And specifically, PacBio has improved a lot recently, because they released this kind of reads, which are called hi-fi reads. These technologies, what they do is to read single molecules of DNA, and what is special about this technology is that they can read the same molecule, that they form this kind of hairpin or circle, and they read the same sequence once, again and again and again and again, and this way the errors that are usually associated with long read sequencing are reduced. So you have a read that is as good as Illumina, but they are longer. So you can have reads that are between 10 and 20 kb, but very accurate, which was a problem before for long read sequencing. So if you have reads that are very, very long and without errors, you can resolve these problems that you will find when you have repetitive sequences. If you have repetitive sequences, you cannot differentiate between these two possibilities, because if you have this sequence is the same as this sequence, and the reads are smaller than the repetitive sequence, you cannot know which fragment comes after the other. But if you have reads that are longer than the repetitive sequence, you can know which sequence come after the other. So now it's much easier to assemble genomes. And this is an example I wanted to show you about the plant that we are working with. Each of these different lines represent a contig, which is a fragment of DNA that was assembled using this technology. So this is an assembly graph using one lane, or one lane of this machine. Lane is the unit that you use for sequencing. This is an assembly graph using 2 lanes. You see that the number of contigs, this is around 3,000 contigs. It decreases to 500 contigs. And then if you use 3 lanes, you have more or less the same number of contigs, but you have contigs that are the same size as chromosomes. You have chromosome scale contigs. So you basically assemble the genome. And this is very easy to do now. I mean, if you have the money, of course, but now there are softwares to do this. So it's not previously to do a new genome, it was a very complex thing to do. Projects that were millions of dollars for doing a new genome, and now this is something that is happening in many labs to sequence a new genome. So why it's an advantage to have a genome? Because you can do transcriptomics much easily, because you won't deal with the problems of alternative splicing. That is also a problem to do transcriptome assembly with short reads. And also you can identify this gene cluster that I was showing before. And these gene clusters allows you to identify new genes that will be probably involved in the pathway that you are looking for. But this technology allows you not only to sequence DNA, you can also sequence RNA, and this, as I told you before, it solves a lot of problems which are related with alternative splicing. When you have alternative splicing, you need that the short reads are located in the regions in exon junction. But if you sequence the whole transcript, the full length transcript, you don't have this problem. You see the whole alternative splicing landscape of this species. So there is no assembly required. So that's it. And I will take any questions that you have.
Thank you, Luis, for this nice presentation. I mean, I think it's better for you to stop your presentation so you can see people. One of the advantages of using this shared screen on Google Meet is that you cannot see other people. So, I mean, if someone had a question that demands you to go back to the presentation, we can go back.
Okay.
So I can open now to questions to everybody. If you want to ask something to Dr. Luis, I can start with one. Can I go first, Magda? Yeah. Okay, Luis, I thought it was very interesting, the approach to identify the whole biosynthetic pathway for those compounds in spinach. But I kind of missed how did the group identify the cellulose synthase as a candidate for this. It was supposed to be a glycosyl transferase, but it was actually a cellulose synthase. And cellulose synthase is a super family. You have cellulose synthases, which are responsible for the biosynthesis of cellulose microfibrils, but you also have all the other cellulose synthase-like families, which are normally involved in the biosynthesis of cell wall polysaccharides, such as hemicellulose and pectins. So it was just because it was co-expressed with this.
I know that they solved all the pathway, but this enzyme was the only one that was missing. And yes, they took it only because of co-expression. Of course, I don't know exactly how many genes they tested, but probably a lot of them. But yes, it was only for co-expression. But this gene had the particularity that it was co-expressed with all the other genes. So it was very strongly co-expressed.
Okay. So it was not like a shot in the dark. It was really targeted. And do you remember to which subfamily of cellulose synthase it belongs?
G.
Okay, G. Yeah, because as I told you, I mean, each of those cellulose synthase subfamilies, they are normally related to Mannan biosynthesis and xyloglucans. There are several of them, and apparently it's conserved across species, right?
Yeah, it's conserved across species. Yes. Yeah.
Okay. Very nice. Okay, thank you, Magda. You can go.
Thank you very much, Luis, for the nice presentation. And I have a couple of questions. The first one is very specific about the glycosylases. You talk about the SOAP 5 to 7, and this is just curiosity, but do they work always in the same sequence? I mean, the different glycosylations are always in the same order. Do you know that? Because this is something very, very common also in the glycosylases in the cell wall. And it's something that I really don't know how it works.
I think most of the enzymes in general are promiscuous. They will not work only with one molecule, but they will work with many molecules. So you can have branches in this pathway. In the paper, in fact, there is a proposed branching of the pathway. I don't have it here, but yes, it's not so linear that as it appears in the figure. This is the question, or?
Yeah. Is that what? So you don't necessarily have all the glycosylases working in the same molecule.
Yeah.
Maybe some residues are missing.
Yeah.
Okay.
Can I make an additional question that is similar to yours? Do they know the subcellular localization of all those enzymes? Because maybe this can tell us what is actually the path in Planta. I mean, because maybe one enzyme, I know that a lot of enzymes involved in these glycosylation steps are connected to the Golgi, for example. So do they know?
Yeah. It's in I didn't put the figure but it's in the paper. It's an endoplasmic reticulum. Yeah. So that's another hint for them to know. And they also tried if this molecule was also involved in micellulose or cellulose synthesis, and nothing it didn't. So yeah.
Okay. And the other question, it's a wider one regarding the workflow that you presented at the beginning.
Yes.
The lab approach. Do you have any kind of approach for metabolite prospection in terms of which is the source of those interesting metabolites that you mentioned at the very beginning? Okay, because tomatine and all that are famous, and I know pretty well, everybody knows them. But what about those interesting new metabolites?
I think for this, the most interesting thing to do is to go to the databases of these companies, like Analyticon, and we don't do this prospection here, but these companies do it. I don't know if that's what you mean.
So Asaf don't used to go to the field to do that because that maybe from native knowledge, you know, and medicinal species use traditionally used, maybe that's a very interesting way to go, and start working with new species or new putative metabolites from that.
I can tell you that now we are starting to work with cacti, and I mean, this is a project I am involved with, but I see how Asaf brought the project, and it's because just he knows that cacti are used by many people in the world for different things, not only for mescaline, which is the compound that is made in San Pedro, or but there is no information about this, about the pathway, so he realized that this not known, so he started to bring cacti, and yes, I think the approach is to try to be with the eyes open to things that are used by humans and the biosynthesis is not known, and then start working on this.
And several plants are traditionally used, and you don't even know which is the active molecule with biological activity. So that's
I think in Brazil is I think the mecca for these kind of things.
Definitely.
Yeah. I think there are labs doing these kind of things in Brazil, no? Or...
Yeah, here in our department there is a relatively big group working with natural compounds and identifying eventual biological activity for new metabolites. Yeah. But they have, I mean, we go outdoors and we find thousands of new plants and identify metabolites everywhere. So here it's really simple. By there, I don't know, I mean, which were the source for these new metabolites, maybe.
Yeah, I think mostly he works on literature. Like once he finds something that interests him, he starts to look in literature, and that's the way. When he finds a plant that produces this metabolite, or it was reported that produces this metabolite, he tries to get it, and that's it.
Thank you very much.
Luis, we have a couple of questions there at YouTube chats. Dr. Cristiane Calisto, she's a professor at our department here at USP. She is asking, how did you generate the gene network that you presented in one of your first slides? I think she's mentioned about the co-expression network.
This, the network of the paper of Adam, this one. I think
you can confirm, because it was the gene network shown in one of your slides. I don't know exactly which one she's mentioning, but
yeah, it was done by Cytoscape. There is a software that was developed in the lab, which is Co-Express BIS. It's a very friendly user software to do co-expression analysis, and this software has a lot of modules to export to softwares like Cytoscape, and this figure was done with first identifying the co-expression with co-express BIS, and then the figure was done with Cytoscape.
I will also take the opportunity. Is the idea of the lab of your lab to make it available? Because I mean, you show like new technologies to sequence genomes, and apparently you're interested in some species that are not like a really model species, but actually that produces interesting compounds. Is it the idea to make these genomes available to the community?
Yeah, my work specifically is mostly related to bioinformatics, to this part of the work, and yes, usually the resources that we published are made available.
Okay. Well, already talking about what you're doing, Daniele Rosado, I think you know, it's a former PhD student at Magda's lab. She's saying that it's a pleasure to hear you talking. And if you can explain a little bit what you're doing exactly, I mean, you don't have to, of course, to tell about your data, but what exactly you're working, and she's sending you a big kiss.
Another one for here. Yes, I'm working with cannabinoid biosynthesis, and we are interested not in the main cannabinoids, not in THC or CBD, but in the minor cannabinoids. There are almost 100 different cannabinoids that you can find either in the cannabis plant, but also in another plant. In different concentrations, of course. So what we are trying to do is try to find genes that will help to increase the variability of cannabinoids, not only these two very big and well-known compounds, which are THC and CBD, but also another ones. So we are trying to find enzymes that will generate variation in the production of cannabinoids. Yes.
Nice. Cool.
Yes.
And is the intention of your life? I know that like people working on metabolomics. I did one postdoc in a lab that was also working a lot on metabolomics, and one of the bottlenecks is always the identification of compounds. Is that part of the lab of Aroni to improve the identification of compounds? I mean, there are several techniques that take like a basic structure that you know the mass, the MZ of this structure, and then you see all the, for example, this is one of the approaches. You see all the chemical modifications that can occur, like this molecule can be glycosylated, can be phosphorylated, can be whatever, and then you have like a network of metabolites that can be further identified and validated using the SMS, of course. Is it part of the aims of your lab to also work on improving the identification of these compounds? Because working on specialized metabolites is like a whole universe of structures. And this is a bottleneck, of course, to identify. Or it's just like identify the compound and that's it. Is it part of the lab to improve these identifications?
Okay, I mean, this is more part of the work of the chemists. There is the lab is divided on more or less chemists, biologists, and bioinformatics. And there are like the paper I showed you about the mass spectrometry imaging. This was also the mass spectrometry imaging in plants was more or less developed here in the lab. Mm-hmm. But I'm not specifically involved in this, but we are using a lot of labeling. I'm not sure if that's what you mean. You know, using labeled compounds to increase the sensitivity of detection of new molecules. I don't know if that's what you mean.
Well, actually, if it's also part of the objectives of the lab to work specifically on the technique. I mean, not like unraveling a new pathway, because this I understood that you guys do, but actually working on the technique itself to improve identifications, and of course this would benefit the whole community of metabolomic people, you know, that, because I mean, I give you an example. I run some samples from sorghum. We were able to detect 15,000 peaks, which then were processed. That could be like something like 12,000 compounds. From 12,000 compounds, we were able to identify 72. So you have 12,000 structures, but you only identify 72. And people believe like doing metabolomics is just like transcriptomics, that you can do a blast and you identify to which gene family that transcripts belong to. And this is not the case with metabolomics, which is very, very complicated. So is it part of the lab to try to help in this sense? I mean, to identify novel structures and publish these and, make a repertory of structures,
I know that there was a paper a few years ago about the database of compounds, but of course, these databases are so dependent on the lab, because even the same machine in different labs will produce different retention times, different. So we have specifically one QTOF machine that we use for metabolomics, and we use every time the same method, the same everything, because this is the only way where we can use the database. So there is a database of the lab. The paper is called I'm looking for it. Weizmann. I will share the link here. So there is a database, but as I told you, it's very. I don't know how this will improve. Yes, how this will improve in terms of reproducibility in different labs, because of these problems of the variability of the machines, the variability of.
Of course.
So I think the lab tries to expand its capacity to detect metabolites, but I don't know if the aim of the lab is to increase universally for everyone. But,
okay.
I know it's a problem.
Yes, it is.
Yeah.
So Magda, you still have your hands up. It's another question. No? Does anyone still want to ask something to Dr. Luis? We don't have more questions in the YouTube chat. Any of the students? The students most likely are shy to ask you something, but yeah, we still can ask him a few questions if you want.
I have one.
Yeah.
It's a curiosity, actually. How about the cannabinoids research? How about the cannabis, the plant issue legally? I mean, in Israel, yeah.
Here in Israel, you know that THC was discovered here in Israel in the 48. So I would say it's very advanced, the law. In terms of medical uses, it's very advanced. And in terms of recreational use, it's almost not prosecuted, the use. So to grow cannabis, like in our institute or in another institute, you have to ask for permission. The police come to see your facility. You have to have some security requirements that you have to comply with. And these requirements, of course, are very expensive. It's like you have to have 24 hours security. You have to have access to the plants very secure, with alarm and everything. But if you comply with everything, it's okay. And the person who works with the plants also doesn't have to have criminal records. And that's it.
Luis, talking about cannabis, do you work with stable genetic transformation or VITs for the functional analysis, or is there?
we are trying to do stable transformation. I try to do it, with these new techniques using developmental regulators. I don't know if you heard about them.
Yeah.
Yeah. So I tried there is a paper with Boitas that was published a year ago or more or less. And I tried to use this technique, but it didn't work, at least for me. But it's I don't have the hand. I think this should be. I told Asaf, this should be a person who really knows tissue culture should work with this. It's not my field. So for up to now, it's a bit stalled these things. But BIGs, yes, there is a paper with the BIGs that worked. We made it work already here in the lab, but it's very. It doesn't go to the whole plant. It goes locally. So it's very complex to work with. Still, we didn't manage to make it go to the flower yet. Yeah, but I think BIGs is BIGs is the most promising technique. Yes, and this is mostly what we are working with. Yeah.
Okay, Magda, go ahead.
Luis, how do you deal with the genetic background and inbreeding? Because, I mean, as far as I know, well, actually, I don't know anything about that, but do you use inbreed lines to have more or less genetic stability?
We got different varieties from that are available in Israel, and I since I came, I picked one of these varieties that I liked because it was very quick very quick for flowering and everything. And this variety, I am selfing it every time I can. And I think we got a very stable, at least phenotypically, very stable plant. Yes, but it's not something that you can generalize. And I didn't do it for many plants. I did it for one that I wanted to be my reference variety.
It's something that you have to work on. I mean, it's not available.
Yeah, in the market, most of the plants or many plants are hybrids. Yes, so if you want to work on genetics, you have to start doing this. Yes, it's not our strong part, but yes, we are doing it.
Okay.
Yeah, but also the problem is that when you do a lot of selfing, you start getting inbred depression. So it has a limit. You cannot go to pure line.
Many generations.
Yeah.
Okay, so you always deal with heterosity.
Yeah, yeah, some degree of heterozygosity is impossible to remove. Yeah.
Luis, before, there's a question from Marcelo Pena at the YouTube, but just before I ask you that question, Magda asked you about this, whether you were like having stable lines and more homozygous. You said that you have several varieties there, and that stable transformation protocol didn't work for any of the varieties, because normally tissue culture is something very connected to the genotype. But even using like 5 or I don't know more varieties, you didn't manage.
We used, I will tell you, we used two varieties. One that was a high THC variety, a regular plant, and another one that was a hemp variety. Very phenotypically very different. Yes, and I didn't manage, but yes, I agree with you. It's a matter of trying different varieties and
different hands.
Yeah, different hands.
I know exactly what you mean. Okay, Marcelo Pena Ferreira is also a colleague of ours at the department. He's thanking you for the presentation, and he's asking you if in the cannabinoid biosynthesis, if you were able to locate the genes responsible for coupling the units from the two biosynthetic pathways.
From the two biosynthetic pathways.
Yeah.
Can I ask to clarify a bit about this? Like,
you mean what the two biosynthetic pathways does he mean? Paulo, if you can complement your question, please. What are the two biosynthetic pathways you were mentioning? I'm not an expert, so I wouldn't know neither to help.
Yeah,
are you mining the genome? I guess for all the biosynthetic pathways, most likely, right?
Sorry?
You are mining the genome towards the identification of all the biosynthetic genes, I guess.
Yeah. Basically, we screen these varieties that we have, and we try to get the ones that are more different, that produce more different cannabinoids. And we start working with we try to focus on these varieties that produce different cannabinoids, and we try to get into these ones to find the new genes. Yeah.
Marcelo clarified there in the chat. It was the pathway dedicated to monoterpenoid. That's one part, and the aromatic part is the other pathway.
Okay, but I think this is known.
All the genes are known in this.
Yeah, I'm looking for the pathway, but I think he means between olivetolic acid and guanylyl diphosphate to generate CBG. But these enzymes are known. Yes, they are yeah. GOT4, it's one of these enzymes that grabs olivetolic acid and GBP and produces CBG. Yeah, okay. We are not focusing on this. We are focusing more on the side chain of the cannabinoids can have different length. Can have usually in the typical cannabinoids it have 5 carbons, and we know that it can have 7, it can have 3, it can have, and these different cannabinoids have very different physiological activities in humans. So we think, and we are working with this. We think it has to do with the availability of short chain fatty acids that will be incorporated to this molecule to produce this C5, C3, or C7.
I think what he meant is that if there's this gene that is responsible for coupling the two units, not exactly the pathway towards the monoterpenic, biosynthetic, and the aromatic, but coupling the units for the structure of the cannabinoids. I think that's the question. I can even paste it here in the chat, and then maybe you can better understand what he meant.
I can share the pathway if you want. Do you see the pathway?
Yeah, but can you put in the presentation mode?
Yes.
Yeah. Okay.
So I think he's asking about this enzyme.
I guess so, yes.
Which it's it mixes the geranyl diphosphate pathway, the monoterpene pathway, with the olivetolic acid pathway to generate CBG, which is the precursor of THCA, CBDA, and CBCA. And this is what we are working now, which is the lipid metabolism to produce different short-chain fatty acids to produce these C5, C3, or C7 cannabinoids.
So everything that is in red there, those are known genes and characteristic enzymes.
Everything in red is already known. Yes, yes. This part is not known. Yes, and we are mostly working on this.
Okay. So you see that I think most of the questions are regarding the cannabinoids. People we have to invite you once again, once you have your paper published, to come and talk about cannabinoids. I have a further question about cannabinoids. Do you have the approach to try to synthesize, like, let's say, unnatural cannabinoids? And I will give an example. The lab of Alain Hussins at VIB, he works on specialized metabolites as well. They had an approach in yeast, so they express, for example, they find out, they discover new pathways for different saponins, and then they can mix in a yeast platform genes from different species that do not occur in the same species, so then they can generate new structures that are not found in nature. Is it possible to do something like this with cannabinoids, instead of just like discovering this C5, C7 to produce, I don't know, like either you can attach some different chemical groups to these cannabinoids to try to produce like a natural, artificial or human-made cannabinoids?
Yes. Do you know Oliver Kaiser?
Yeah.
Okay. Oliver Kaiser is doing this, and also I missed the name. There is Kingsley. Kingsley in the US.
California?
California.
Oh,
yes.
yeah.
Yeah, he has a paper that was published, I think, a year ago, and they did natural and unnatural homologs of cannabinoids in yeast. Yes.
Cool.
Yeah. We are not working on this, but yes,
it's possible. Okay. Nice. Let me check if there's any other questions. Well, I think not. Neither here in the chat. So, anyone else?
I want to thank you very much, Luis, because I have another meeting right now, so I have to leave you guys. But thank you very, very much for your presentation, and it was a pleasure to be with you for a while now. So take care, and we keep in touch. And see you, everybody, and take care, everybody, until Thursday.
Okay, bye, Magda.
Thank you.
Actually, I think we don't have any more questions, so I would like to thank you, Luis. In the name of the whole team of the course, Leandro, Magda, and I, and in the name of the students, it was very nice to hear your talk again. And as I said, we are expecting, don't be surprised if we invite you in the future. I will follow your publications. Once that's cannabinoid work is published, we can invite you again, and then you can give a talk about that as well. So thank you very much, and we keep in touch.
We keep in touch.
Thank you.
Bye.